Hello, my name is Amber Mills, and I'm currently a postdoctoral scholar at the University of Pittsburgh. But this research was conducted during my time pursuing a PhD at West Virginia University under the leadership of Dr. Mark Olfert. During my time at WVU, I was highly interested in the effect of maternal e-cigarette exposure or electronic cigarette exposure, more commonly known as vaping, on offspring vascular health. The use of e-cigarette products have been used to replace cigarettes, and the harms of cigarettes are well documented. However, this switch has come without proper data showing e-cigarette efficacy and safety. Over 10 years later, there are numerous publications demonstrating the harms of e-cigarette use. And despite this, there are still agencies that promote use of e-cigarettes even during pregnancy. Prior published data from our lab has shown that exposure to e-cigarettes during pregnancy leads to poor vascular outcomes in the offspring, and that gestation only and not lactation are the main drivers of cerebrovascular impairment in offspring. To further isolate the critical window during pregnancy, we exposed female Sprigdali rats to e-cigarettes either during their first, second, or third trimester. And then we compared that to a full-term exposure. And additionally, we also did an exposure 3 weeks before mating, also known as a preconception exposure. And then we compared those to our controls, which were exposed to air. We studied the offspring at weaning, which is 3 weeks after the offspring are born, and then at 3 months to get an adolescent time point, and 6 months to look at an adult time point. We found that late in gestation, around 2 to 3 trimester area, is when adolescents and adult offspring age suffer cerebrovascular deficits while using pressure myography in the middle cerebral artery. We also noticed anxiety-like behaviors using the behavior test open field. Additionally, there was increase in fetal loss both during gestation and also during the weaning period. So here you can see the offspring have decreasing MCA function in response to acetylcholine as the exposure to e-cigarettes is later in gestation, with second, third, and full-term gestational exposure being significantly different compared to controls. And this trend persisted in both males and females and at both 3 and 6 months of age. And as mentioned, these offspring also had a decrease in central tendency, which is a known anxiety-like behavior in offspring. And we saw this at the adolescent time point. In addition to that, we also saw offspring loss during gestation in the second, third, and full gestational exposed offspring. And we saw offspring loss both during gestation, which would be reabsorptions, but also during the weaning period. And so here you can see the number of offspring that were born versus the number of offspring that made it to weaning. And you can see that for the second, third, and full trimester exposures, we did see significant loss in offspring as they were being weaned. This paper is the first to characterize the significance of gestational exposure with e-cigarettes and adds to the literature that vaping is harmful. It's a great launching point for others to do more specific investigations into the timing of the harm to offspring and other physiological parameters such as brain and lung development. I want to thank you for taking the time to learn more about our published study in the Journal of Physiology. The full paper was published in volume 602, issue 17, and please contact Mark Olfert at imolfert@hsc.wvu.edu if you have any further questions. Thanks.